foundation for the DNA as a genetic material. Griffith only confirmed that there is going to be transformation occurs. It does not confirm which biomolecule is going to be transformed. Therefore, the updated experiment is going to be Avery Mcleod McCarthy experiment from Griffith experiment. Here, they uses a Griffith transformation principle itself and they undergo further updation to gather the evidence for the transformation principle to identify which is going to be a genetic material. Therefore, Griffith could understand the cause of a bacterial transformation. It was first proposed by Oswald Avery, Mcleod and Mcleod McCarthy. These three scientists are going to propose this experiment from the Griffith experiment. So we can tell that it was a purified form of transformation principle. So here the first step is going to be they prepare these samples of DNA from S3 bacteria, and also the R bacterial cells are going to be prepared and they were cultured off in the agar. Then the S3 bacteria culture are going to be introduced into the R live bacterial culture. R live bacterial colonies. When the S3 bacteria are going to be introduced into the R bacteria, the resulting colony, when it was introduced, lead to the R live bacteria will become S bacteria, which means the rough strain bacteria culture became the smooth culture due to the introduction of smooth culture. Here we can find that a smooth culture, that is smooth strain bacteria, convert the rough strain bacteria also into smooth strain. So the R2 colonies arise from the original mixture is also taken off and grown separately. When the introduction of the S strain bacteria into the rough strain bacteria, it causes the transformation of rough strain into S strain. So here the transformation can be found not from the live source, it is done by the in vitro process in the cultures, so that we can find the image representation more clearly how the transformation is going to be taken place. Due to the presence of S strain, there is a transformation of R strain to S strain is taken place. So here we have to find which is going to be transferred. So the four method is going to be used for analysis. First is going to be chemical analysis. There is a certain chemical reaction to be taken place. This chemical reaction confirms that the presence of deoxyribose in the transformed component. And also by physical analysis we can find. In the physical analysis it is found that a viscous substance is going to be DNA to be physically found. Therefore, when the sample was found physically to be viscous, it was confirmed that it is going to be a DNA. Then a transformation principle can also be identified with the analysis of suspected components are going to be RNA, protein, lipids or glucose. So the found material, found culture are going to be treated with proteolytic enzymes like trypsin and chymotrypsin. If the genetic material is going to be a protein, it causes lysis by trypsin and chymotrypsin. And also it was treated with RNAs. If it is RNA, it can be lead to lack of transformation principle after treatment with RNAs. If then it was treated with lipase. When it was a genetic material is lipid, it causes the lack of transformation principle after the lipase introduction to the components. But even it was treated with trypsin, chymotrypsin, RNAs and lipase, there is no lack of transformation principle. That means there is the presence of transformation principle after the treatment of these components, after this treatment of these enzymes. So it was found that the genetic material can't be a protein, can't be a RNA, can't be a lipid. So to confirm that it was a DNA, the culture is then treated with DNase enzyme. When it was treated with the DNase enzyme, which can break off the DNA, when it was treated with the DNase, it causes the inactivation of transformation principle. So it was confirmed that the DNA is going to be found as a genetic material. So here we can find it is a heat-killed H strain bacteria from the culture. Heat-killed H strain and R strain bacteria from the culture. To identify the component which is going to be a genetic material, was treated with the proteinase enzyme, which is going to be chymotrypsin or trypsin, or going to be used when the proteinase enzyme is introduced in this H strain. It will lead to broken down of proteins. After that too causes the R strain to be transformed. That means a transformation is going to be happen after the proteinase introduction also. Then RNase is introduced. So the RNase is introduced, the transformation happens. So here we can find it is not RNA, not a protein. Even a lipase is also introduced. It too causes a transformation, which shows that there was no lipid is going to be a genetic material. So when the DNase enzyme is introduced, it causes the lack of transformation. So it was found that DNA is a transforming factor, which is a genetic material to transform from one bacteria to another, one cell to another, the characteristics of H strain bacteria. Hence, DNA is a transforming factor and genetic material was found by Avery Mcleod McCarthy experiment.